Welcome back. Physics builds models. Chemistry builds structures. Biology builds evolutions. 3 ladders, 1 way of thinking. And today, we drop that thinking straight onto a real exam idea: 2018 VCE Biology NHT DNA manipulation techniques and applications question set. Today's focus: CRISPR, Cas9, gRNA targeting, double-strand breaks, and DNA repair pathways. A group of scientists is seeking approval to edit the DNA of human embryos to discover which genes play a key role during an embryo's first week of development. The scientists plan to use only 1-day-old embryos donated for research purposes. A total of 25 embryos will be required for the study. A similar study in another country used fewer than 10 embryos. What is the advantage of using 25 embryos instead of 10 embryos? 1 mark. Conflating reliability or precision with accuracy. Increasing the sample size, from 10 to 25, reduces the effect of random errors and increases the reliability and precision of the average outcome. It does not inherently increase accuracy, how close a measurement is to the true value, as sample size alone does not correct for systematic bias or calibration errors. Confusing repetition with sample size. Increasing sample size, equals 25, is replication, whereas repeating the experiment is reproducibility or repeatability. For a 1-mark question asking for an advantage of a larger sample size, equals 25 versus 10, VCAA assessors typically accept any one of the following distinct points. Option 1, impact of outliers and errors. Increases the reliability of the results by reducing the impact of random errors or outliers on the data. Option 2, biological or genetic variance. Better accounts for natural genetic variation among human embryos, ensuring the findings are more representative. Option 3, precision and replicability. Increases the precision and statistical power of the experiment to detect true biological effects. The scientists will edit, target and change genes in the embryos by switching off genes that are thought to be involved with the earliest stages of development. Embryo growth will then be observed over the next 3 days. What results would support the hypothesis that is being tested by the scientists? 1 mark. Deconstructing the question logic. The scenario describes a classic loss-of-function knockout experiment. Independent variable, 4. Editing or switching off specific target genes in the embryo. Dependent variable, DV. Observation of embryonic growth and development over 3 days. Tested hypothesis: the targeted genes play a key, essential role in early embryo development. To support this hypothesis, disabling the targeted genes must result in a failure of early development compared to normal growth. Logical ladders. Cause and effect relationship. The response explicitly connects the manipulation of the 4, knocking out or switching off the gene, to the outcome in the DV, arrested development, a failure to function. Comparison baseline, implicit control. For full marks in VCE questions like this, answers should clearly state what is observed in the experimental group, e.g., development stops, growth ceases, or cell division fails, compared to normal expectations. High-scoring response options. For a 1-mark question asking, what results would support the hypothesis, any of the following concise formulations will secure the mark. Option 1, direct observation. Embryos with the switched-off gene cease to grow, fail to divide, or show abnormal early development. Option 2, loss of function outcome. If knocking out the target gene stops or impairs embryonic development over the 3 days, it indicates those genes are essential for early development. Option 3, comparative response. The edited embryos exhibit significantly reduced growth or developmental arrest compared to unedited, control embryos. The CRISPR technique is a gene editing method. It involves a protein called Cas9 and a short piece of guide RNA, gRNA. The gRNA leads Cas9 to a gene in the DNA that scientists wish to edit. Cas9 is an endonuclease that acts in a similar way to a restriction enzyme. C. What action is Cas9 expected to perform? 1 mark. Overview. The CRISPR Cas9 complex. The CRISPR Cas9 system is a powerful molecular technology used for precise gene editing. Its core components are shown in the CRISPR Cas9 complex diagram, top left. The system consists of 2 main elements. Cas9 protein, a dark blue, oval-shaped enzyme that functions as molecular scissors. Guide RNA, gRNA, a single-stranded RNA molecule, shown in red and orange. The specific sequence at the 5' end of the gRNA, the CRISPR target sequence, highlighted in red, is what gives the system its precision. It is engineered by scientists to match and bind a complementary target DNA sequence in the genome. The 3' end of the gRNA contains a scaffold sequence that anchors the gRNA within the Cas9 protein. 1. Complex formation and target recognition. The system consists of the Cas9 enzyme, molecular scissors, bound to a guide RNA, gRNA. The gRNA contains a sequence engineered to match the target DNA site. The complex scans the genome, binds to a PAM sequence, and the gRNA hybridizes with the complementary target DNA strand. 2. DNA cleavage. The Cas9 endonuclease domains cut both strands of the target DNA, creating a double-strand break, DSB. 3. Repair pathways and outcomes. Non-homologous end joining, NHEJ. An error-prone repair pathway that directly glues the ends together, often introducing random insertions or deletions, indels. This causes frameshift mutations, resulting in a gene knockout, gene inactivation. Homology-directed repair, HDR. A precise repair pathway that uses an added donor DNA template, with matching homology arms. The cell incorporates the template sequence during repair, enabling gene correction, or knock-in gene insertion. One. Key biochemical function. The stem explicitly sets up the analogy by calling Cas9 an endonuclease that acts like a restriction enzyme. Primary function: cleaving the phosphodiester backbone of target double-stranded DNA. Mechanism: it makes a double-strand break, DSB, at a specific site targeted by the complementary gRNA sequence. 2. Examination key point breakdown. The mark scheme states: acts as molecular scissors that cut the DNA strands at a specific sequence or location. To ensure students get the full 1 mark without ambiguity, they must mention the action, cuts, cleaves, or edits the DNA of phosphodiester bonds. The target, DNA at a specific site or location, guided by the gRNA. 3. Ideal student responses. Concise response. Cas9 acts as an endonuclease to cut or cleave double-stranded DNA at a specific target site. Alternative formal response. It cleaves the phosphodiester backbone of the target gene's DNA sequence, creating a double-strand break. 4. Common student pitfalls. Attributing specificity to Cas9 alone, students often forget to clarify that Cas9 is guided by gRNA. Cas9 itself provides the catalytic cutting function, gRNA provides sequence specificity. Vague verbiage, simply saying it edits the DNA, or it removes the gene, is usually too imprecise for VCAA. The exact mechanical action, cutting or cleaving, is required. DNA ligase is required for the next stage of the editing process. What role would DNA ligase play? 1 mark. DNA ligase acts as the molecular glue in the final stage of CRISPR Cas9 gene editing by sealing the double-strand breaks created by Cas9 to permanently reform the DNA backbone. Concise response. DNA ligase joins the DNA strands back together after the editing or insertion is complete. Biochemically precise response. It catalyzes the formation of phosphodiester bonds to seal the sugar phosphate backbone, joining the broken DNA strands, or integrating the new DNA fragment. Common student pitfalls and teaching tips. When prepping students for questions on DNA ligase, several classic conceptual errors tend to surface under exam conditions. Confusing phosphodiester versus hydrogen bonds. A frequent error is stating that ligase joins complementary base pairs, or forms hydrogen bonds between the 2 DNA strands. Students must remember that complementary bases pair spontaneously via hydrogen bonding. Ligase is strictly responsible for covalent bonding along the vertical sugar phosphate backbone. Conflating ligase with polymerase. Students sometimes write that ligase adds nucleotides to fill the gap. It is crucial to reinforce the division of labor. DNA polymerase synthesizes and adds new nucleotides, whereas DNA ligase only seals nicks and joins existing ends. Vague terminology. Responses like "it fixes the DNA" or "it glues the gene" are routinely penalized. Students should be coached to use mechanical action verbs, joins, seals, connects, ligates, coupled with the correct anatomical target, strands of fragments, a sugar phosphate backbone. The CRISPR technique can be used to insert new genes, or to delete sections of a gene. While some scientists have recognized the potential benefits of using the CRISPR technique to edit genes in the future, other scientists have expressed concern. State one potential benefit some scientists may have recognized and one concern other scientists may have had. 2 marks. Because the broader scenario in question 9 involves editing human embryos, responses must align with the implications of germline gene editing rather than just somatic editing. Benefit, 1 mark. Focuses on the therapeutic or medical potential of modifying embryonic DNA. Concern, 1 mark. Focuses on the biological risks, e.g., off-target mutations, inheritable changes, or ethical dilemmas, e.g., destruction of embryos, consent, designer babies. Potential benefits, choose ONE. Elimination of inherited disorders. Ability to correct disease-causing mutations, such as Huntington's disease, cystic fibrosis, at the embryonic stage so the child is born free of the condition. Improving fertility, IVF success. Identifying and editing genes critical to early embryonic development to improve implantation and successful pregnancy rates in IVF. Potential concerns, choose ONE. Off-target mutations or unintended errors. Cas9 may cut unintended sites in the genome, causing harmful mutations, or inadvertently introducing new diseases into the embryo's DNA. Inheritable germline changes. Changes made to embryonic DNA will affect all future cells, including germ cells, sperm, egg, meaning unintended mutations, or edits are permanently passed on to future generations. Ethical issues with embryo research. The destruction of human embryos during research raises moral and ethical concerns regarding the sanctity of potential human life. Societal and ethical slippery slope. Concerns around designer babies or non-therapeutic enhancements leading to social inequality. High-scoring exam responses. To guarantee full marks, 1 mark for benefit, 1 mark for concern, students should write clear, complete sentences anchored directly in the context. Benefit. It allows scientists to permanently correct and eliminate life-threatening genetic diseases from an embryo before the child is born. Concern. Unintended off-target edits could introduce dangerous mutations into the embryonic DNA, which would then be permanently inherited by future generations. Key teaching points for students. Distinguish germline versus somatic editing. Remind students that editing embryos alters every cell in the resulting individual, including their gametes. This makes the edits inheritable, which is a primary source of ethical concern. Precision in concern terminology. Encourage students to use technical bioethical terms where applicable, such as off-target effects, germline modification, or gene pool consequences, rather than vague statements like "it might go wrong." The scientists will need to make multiple copies of gRNA to place into each embryo. Polymerase chain reaction, PCR, will be performed to produce many copies of the DNA template sequence that is used to create the gRNA molecules. Two different primers are used in the copying process. Explain the role played by primers and why it is necessary to have two different primers. 3 marks. Breakdown of mark allocation, 3 marks. To secure all 3 marks on this classic PCR question, the response must address both parts of the prompt, the role of primers, and why two different primers are necessary. Mark 1. Role of primers bind or anneal to complementary single-stranded DNA sequence. Mark 2. Role of primers provide a free 3' end for SAQ DNA polymerase to attach and initiate elongation. Mark 3. Why two primers target opposite ends of different sequence strands, 5' to 3 synthesis directionality. Mark 1 and 2. The biochemical role of primers annealing a specificity. Primers are short single-stranded nucleic acid sequences that bind to target single-stranded DNA via complementary base pairing. Polymerase initiation, DNA polymerase, such as SAQ polymerase, cannot synthesize DNA de novo. It requires a pre-existing 3' hydroxyl group to add free DNTPs. The primer creates this starting point. Mark 3. Why TWO different primers are needed. This is a standard VCAA trick where students give half an answer, because DNA has 2 strands. A high-scoring response requires 2 distinct reasons linked together. Sequence difference. DNA strands are antiparallel with different nucleotide sequences at the 3' ends of the target region, forward versus reverse target regions. Directionality of polymerase. DNA polymerase only synthesizes DNA in the 5' to 3 direction. To amplify both separated strands simultaneously, one primer, forward, must anneal to the 3' end of the template strand, and a second primer, reverse, must anneal to the 3' end of the coding strand. Exemplar high-scoring answer. Role of primers, 2 marks. Primers anneal to complementary target sequences on single-stranded DNA, providing a starting point, free 3' end, for SAQ DNA polymerase to attach and begin extension synthesis. Why 2 primers are necessary, 1 mark. 2 different primers are needed because the 2 DNA template strands have different nucleotide sequences and run antiparallel. Since DNA polymerase only synthesizes in the 5' direction, a forward and reverse primer are required to amplify both strands. Key teaching and exam tips for students. Avoid vague language. Saying primers start PCR, or primers tell polymerase where to go, is too informal for VCAA. Encourage students to explicitly mention annealing via complementary base pairing and providing an attachment site, free 3' end. Highlight antiparallel and directionality. Remind students that stating DNA is double-stranded is only half the mark for the second part. The key reason 2 different primers are needed is that the 3 target sequence on the top strand is not identical to the 3 target sequence on the bottom strand. Experimental design and sample size. Using 25 embryos increases reliability by reducing random error and capturing natural genetic variation. Increasing the sample size from 10 to 25 reduces the effect of random errors. Testing gene function in early development. Hypothesis supported: if edited embryos fail to grow, a divide compared to normal controls. Embryos with the switched-off gene cease to grow, fail to divide. CRISPR mechanism. Cas9 equals molecular scissors, makes a double-strand break at a gRNA-targeted site. Cas9 acts as an endonuclease to cut or cleave double-stranded DNA. DNA ligase seals the break by forming phosphodiester bonds. DNA ligase joins the DNA strands back together, sealing the sugar phosphate backbone. Benefits and concerns of embryo gene editing. Benefit: correcting inherited diseases at the embryonic stage. Permanently correct and eliminate life-threatening genetic diseases. Concern: off-target mutations and inheritable germline changes. Unintended off-target edits, permanently inherited by future generations. PCR and primers. Primers anneal to target DNA and provide a free 3-foot end for polymerase. Primers anneal, providing a starting point, free 3-foot end, for Taq polymerase. 2 primers are needed because DNA strands are antiparallel and polymerase works 5-3. 2 different primers, strands have different nucleotide sequences and run antiparallel. At the end of every lesson, I want to leave you with one idea: learning becomes powerful when you climb the logic ladder. Physics moves from situations to models to equations. Chemistry builds from structure to property to reaction. Biology grows from variation to adaptation to survival. Different subjects, but the same underlying logic. When you learn to decode systems, build ladders, and construct logic, you don't just solve problems, you understand how they fit together. If you'd like to keep climbing this ladder with me, you can find more lessons, breakdowns, and reconstructions on my channel. Thanks for learning with me today.